talk on. Thank you.
Okay, thanks, Laura. So just quickly an announcement about our division meeting 2 weeks from now. We're going to have some clinical updates on all the operations and how they've been affected by COVID. Grant will talk in Ingeborg. There's a lot of changes going on in Roosevelt. And we hope to have somebody from Northwest attend. But if there are other items you want, we have other items, but if you want to send Laura anything that needs to be discussed at this meeting in 2 weeks, please send the items to Laura. Okay, so this morning I'm delighted to introduce our newest faculty recruit, Chris Sims. Dr. Sims received his MD from Johns Hopkins University, was a fellow at Stanford, and for the last 12 years or so has worked at the University of North Carolina, Chapel Hill. At UNC, Chris had dual appointments. He was clinical professor in the Division of Rheumatology. He's seeing patients weekly, so he's a practicing rheumatologist. And he also held a position as research professor in the Department of Chemistry. As you will hear shortly, his research is multidisciplinary, using techniques from chemistry, engineering, and medicine, and his work has a decided translational focus. In his lab description, Chris lists 3 areas of active interest: analytical techniques for single-cell biochemical analysis, microfabricated platforms for isolating single cells for analysis, and the organ-on-a-chip system for recapitulating organ level function. He has over 100 publications, a dozen issued patents. He's the founder of several companies, not to mention very deep NIH grant support. So, Chris, we welcome you to the division and look forward to hearing your talk.
Well, thank you, Keith. I have to say it's been an interesting few months here at Seattle. I'm looking forward to meeting everyone. Hello, everyone. Let me see if I can just get my system going here. Let me know, Keith, that you can see my my screen. There we go. Does that look good for you, Keith?
I don't see your screen, Chris.
Okay, let's see where the problem lies. Let me just technology. It's a
yeah. That green thing at the bottom where it says share screen.
Yeah, I know that. I'm trying to bring up my.
Yeah, you have to pick the.
Exact... there we go. Okay, now you should see it. Does that look good?
Yeah, great.
Okay, terrific. Well, let me start again here. Okay, so I guess I am a conflicted individual, so people tell me. And the reason for that is that, you know, I work with a group that is a technology development group, and one of my roles in the lab is to translate the technologies that we develop out of the academia into industry. And so feel free to take everything I say with a grain of salt, which if you knew me well, you would think would probably be a good idea anyway. So I'd like to start off my talks with giving credit where credit is due. As I mentioned, you know, I'm part of a team. It's a highly interdisciplinary team. We've got engineers, chemists, physicists, computer science folks. So it's a very energetic group, and it's run by a quarterback by the name of Nancy Albritton. Some of you may know Nancy. She's the new dean of the College of Engineering. And Nancy and I have worked together for, now more than 25 years. In the interest of full disclosure, she and I have been married even longer. But it's really other people that do a lot of the work. You know, it's been more than 15 years since I've wielded a pipette man in anger. And this is the group that, you know, really does the work. Our staff scientists, postdocs, graduate students, undergraduates, great group to work with, always keep me on my toes. I particularly want to point out this individual in the middle here with the blue shirt, Yuli Wang. Yuli has worked with us for over a decade now, and he's just an incredible experimentalist. So a lot of our papers, you'll see Yuli as the first author on. You know, when I started putting this talk together, one of the challenges that I had was trying to make it as relevant as possible to the current audience. Ironically, although I'm a card-carrying rheumatologist, a lot of our work does not involve the clinical science of rheumatology. So what I thought I would do is to give you a kind of a high-level overview of 3 of the ongoing projects in the laboratory to try and give you some insight into kind of work that we do, the technology development, and how those technologies might be useful to you in your own work. And more excitingly, if you can think of new technologies that we could work together and create that would allow you to do things that you can't currently do, that's really where we like to go. We have a lot of collaborative work. We had a very rich collaborative network at UNC where we were before, and we hope to build that kind of network here at UW, and I'm sure we will be able to do so. So the 3 projects that we'll talk about, one is our in vitro tissue models. You may have heard of organ on chip, like heart on chip, brain on chip. We work on intestine. Another area, which is completely separate from that, is our microfabricated arrays for doing cell sorting. I'll touch bases briefly on that during the talk. And I'll start off with some work in single-cell enzyme assays. This is a topic that is near and dear to my heart because when I left clinical medicine back in the early 90s to become a bench scientist, this was one of the first projects that I took on. The project is still going on today. That may give you some idea of some of the timelines for technology development. And when I first started this, probably the most common question I got was, why bother doing single cells? I think since that time, that answer has become quite obvious, particularly in the area of tumor cell biology, but also other types of cell biology. In tumors, you know, the tumor cells themselves may only be a minority of a tumor in a patient, the majority often being stromal and inflammatory cells. But more importantly, I think with the advent of single-cell RNA sequencing, we now know that within a tumor, different regions may have different subclones with different and unique genetic signatures. So it's important to do single cells to be able to understand the tumor cell biology. I think for this, currently, the question that is maybe more relevant is, why bother doing enzyme activity? And my argument here is that you know, cellular function is inherently an enzymatic process. You have all of these signal transduction enzymes that are driving cells to behave in particular ways. And it would be very good to be able to actually measure that activity as opposed to what we primarily do now, which is to look at genetic mutations and expression levels and infer the activity based on those findings. But you know, mRNA and protein levels are not direct measures of protein function, and many of our targeted therapies, particularly the small molecule inhibitors, are directed at the catalytic site of particular enzymes, proteins that are signal transduction proteins and other types in the cell. So the approach that we've taken to do this is pretty straightforward. We essentially use a single cell as its own test tube, so to speak. So let me just walk you through this. We have substrates for an enzyme, just like you might have in a test tube in your lab. And here shown, the substrate is a small peptide that has a phosphorylatable residue for measuring protein kinase activity. Now we take, I don't know why there it is. We tag our substrate with a fluorophore, and more on that in just a minute. And we've used a variety of different types of substrates depending on the enzyme that we want to, whose activity we want to measure. Right now, we're doing a lot of work in the area of lipids and lipid signaling, but we can develop reporters for a variety of different types of enzymatic activity. So what we do is we load this reporter into a cell by any number of means, let the protein in that cell act on our reporter. Then we lyse the cell using a pulsed laser, which essentially releases all the cell contents. We collect those cell contents into a capillary and subject it to an electric field, meaning electrophoresis, and then we detect the compound by its fluorescent tag using laser-induced fluorescence detection in a capillary. So going back to our cell that we've loaded, if the enzyme is inactive in the cell, we lyse the cell, collect its contents, electrophoresis. On our electrophoregram, we see one peak that corresponds to our substrate. But if, on the other hand, our enzyme is active, some or all of that substrate. A couple of things that's important. One is we know that the identity of these peaks based on their migration time in the capillary. And also, this process is quantitative. We can tell you the exact amount of moles in each of these peaks so that we can determine the amount of the substrate that is converted to product. Usually how we report the data is percentage of substrate that has been converted into its product. So that's pretty straightforward. Developing the technology to do so has been a little bit more tricky. Basically, we have a microscope so that we can see the cells that we're interested in measuring. In that microscope, we've integrated the pulse laser that I mentioned before. This is a neodymium YAG laser that puts out a 5 nanosecond pulse, and that causes lysis of the cell when it's directed on the cell. And then we build a laser-induced fluorescence detection scheme for that capillary that gives us a very high sensitivity to allow us to measure very scant amounts of our reporter when loaded into a cell. So hopefully this animation that one of our graduate students did will work well. You'll see the microscope, it'll turn yellow, there we go. And then, okay, I've been having trouble getting this, there we go. And then this is our laser-induced fluorescence detection scheme hooked to the microscope. We're gonna hone in on the inlet of the capillary, which is positioned above a cell, in this case on a cell array, although that's not that necessary. And it's in buffer, the cells are alive, they've been loaded with our reporter. The pulse laser comes in and blows up the cell. The cell contents are sucked up into the capillary where they're electrophoresed, and downstream there's a window in the capillary that's interrogated by CW laser, and the fluorophores on the substrate emit when they come by and give us our typical data output, which is the 2 peaks I mentioned before about substrate and product. So you can imagine that it's really important to have reporters that actually report what you want them to. A lot of work has gone into this. Well over an hour-long talk could be done on our reporter development. Suffice it to say here that we've developed reporters for serine threonine and tyrosine kinases. We've developed a number of lipid-based reporters and even substrates for the proteasome to look at proteasome activity in multiple myeloma cells. And we've worked with a lot of organic chemists and clinical scientists in developing these various reporter and validating them. Another aspect, particularly with single cell technologies, is throughput. You know, when I first started this technique back some years ago, a really, really good day, I'd be able to analyze 10 cells. The problem is many of the experiments that you need to run have to generate many more cells to get statistically meaningful data. So the approach that we've taken to do this is twofold. One, we've made the system automated, so we don't have to do all this by hand. And the other is to serially analyze the cells. So we lyse one cell, load it into the capillary, followed by another, by another, by another, and that generates what looks like a fairly complicated electrophoregram shown here, where fluorescence output from each cell is a peak, and the x-axis is the time in the capillary to reach the detection window. But it's actually pretty straightforward, and we have computer algorithms that can analyze this data, so we don't have to do this by hand. But if we take one small section of the electrophoregram and blow it up, you can see that this is just a series of peak pairs coming from 5 different cells. Now, it's important to note here, this was a test system for the for the technology. The cells here have been just loaded with 2 different fluorophores, so you get nice clean peak pairs coming out of each cell. And in real world data, the heterogeneity between cells is much greater, which I'll show you in just a moment. So one of the examples for this technology is looking at the activity of sphingosine kinase. Sphingosine is a lipid. It exists in an important signal transduction pathway in what's called the sphingosine rheostat. The molecule sphingosine can be phosphorylated by sphingosine kinase. Oops, going the wrong way. And if it gets phosphorylated, it drives the cell down a pathway of proliferation and survival. That's something that you want to see in cancer. I should have mentioned that this is work we're doing with a collaborator looking at sphingosine kinase activity in acute myelogenous leukemia. The sphingosine kinase inhibitor fingolimod is actually used in multiple sclerosis, but as far as I can tell, that's the only autoimmune disease that it's being used in. It doesn't seem to have been efficacious in our rheumatic diseases. In any case, back to our scheme here. If instead sphingosine is converted to ceramide by ceramide synthase, it drives the cell into apoptosis. So that's something you'd like to see in cancer. So what we've done is our collaborator has given us the peripheral blood blasts from 4 patients with AML in blast crisis, and we have measured the sphingosine kinase activity in those cells. This is shown here as the electrophoregram from cells from one of those patients. And you can see in the blow up that there are 5 cells present. And if you look at the cell number 1, below that is a series of peaks. Our reporter, which is here, sphingosine fluorescein. So it's a sphingosine that's been labeled with fluorescein. And then a small peak for sphingosine 1 phosphate. So a small amount of that reporter has been phosphorylated. But if you look at the other 4 cells, all of the substrate has been pretty much converted into the product sphingosine 1 phosphate. Now, you note there's a 3rd peak here, HAF, which took us a long time to figure out what that peak was. It turns out that it is hexadecanoic acid, which is a downstream metabolic product of sphingosine 1 phosphate. And that points out an advantage and a challenge for our technology, and that is the advantage is we can measure multiple metabolic products and metabolic pathways in a single cell, although it does make our analysis scheme a little bit more challenging. So what kind of information did we glean from these 4 subjects? I just want to preface by saying these are very few subjects. It's hard to make any clinically relevant conclusions from this, but I think it illustrates the potential for a technology like this. The first 2 subjects, 1 and 2, had very little phosphorylation of our reporter, as shown in these histograms, with the x-axis showing the percent of phosphorylation of the reporter. But subjects 3 and 4 had significantly greater sphingosine kinase activity. So basically we could separate these 4 patients into 2 groups with low and high sphingosine kinase activity. And you could imagine using a technology like this to determine maybe the best approach for tailoring therapy to your patient population, where subject 1 and subject 2, you might not want to use a sphingosine kinase inhibitor, but that might be effective therapy for subjects 3 and 4. This is ongoing work, and we're early days yet. We have to do many more subjects to try and come up with clinically meaningful data, but I think it's illustrative. Interestingly, in one of those 2 high phosphorylation state patients, taking their cells ex vivo and exposing them to a sphingosine kinase inhibitor that's being used in clinical trials, we saw that the vast majority of cells, 82% on our dot plot here, had their sphingosine kinase activity inhibited, but there was a small but significant population, subpopulation of 18% that was still phosphorylating the substrate reporter. So you might use an approach like this to either adjust the dose in this patient, or try out a different Sphingosine kinase inhibitor, or use a cocktail therapy. So again, the possibility of tailoring therapy based on the output from an assay of this sort. So we have done one series of experiments related to rheumatoid arthritis. Worked with a collaborator who had a biobank of primary synovial fibroblasts from patients with rheumatoid arthritis and osteoarthritis, and she was interested in looking at the activity of protein kinase B, also known as AKT, in these synovial fibroblasts. When we looked at individual cells from 3 subjects with osteoarthritis, we saw very little AKT activity. I should preface this by saying that these are cells that have been serum starved overnight, and so their baseline activity is very low. But in the RA patients, 2 of the 3 patients had significant amounts of significant numbers of cells that had elevated AKT activity. Now, what's the clinical meaning of this? We don't know at this point in time. Unfortunately, our collaborator moved to a different institution, and our graduate student graduated, and so the project has kind of sat on the back burner ever since then. Interesting to note, when we looked at these cells by Western blot analysis, you could not pick out these differences between the 2 groups, pointing out to the potential value of single cell analysis in these kinds of studies. Okay, to change gears to our microfabricated arrays for cell sorting. I don't think I need to explain to this group that cell sorting technologies are important in cell biology. 2 of the common ways to sort cells are fluorescence activated cell sorting, or FACS, and limiting dilution. FACS sorting is an incredibly powerful technology, very, very high throughput. Some of the drawbacks are the FACS sorter is a fairly expensive instrument. More pertinently, perhaps, is that especially for primary cells, many other cell types, they tend to die from the stresses from FACS, and so it's difficult to generate viable cells from the FACS sort. Limiting dilution is much more gentle on the cells. It's a process by which you take a suspension of cells and dilute it, then take a volume of that by Poisson statistics is predicted to have 0 or 1 cells in that volume, and you put that into generally a 96 well plate, and you can clone your cells in that manner. Inexpensive, manpower, and intensive and tedious. So we came up with a completely different approach to cell sorting. We call them Microraft arrays, and the reason we call them raft arrays will become obvious in just a minute. So what we do is we take a microwell array that has anywhere from 15 to 45,000 wells in a 1 inch by 1 inch region, and we take our cells and plate them similarly to limiting dilution. We put fewer cells down than we have sites. So by Poisson distributions, we'll have either 0 or 1 cell in any well. Then we can look at the array under a microscope and choose our target cells based on any microscopic signature that you can think of. We're able to remove the cells, collect them, and they're highly viable, essentially 100% viability after collection, and we can expand them for cloning or use them for other single cell analyses. RNA sequencing of single cells has been highly popular over recent years. So let me hone in on the technology a little bit more. In the bottom left panel here, you see a very small section of these microwell arrays. Each square is 1 microwell. The unique aspects of their system is that each well contains a little disc of polystyrene, the same material that's used for your tissue culture flasks. The unique aspect of our polystyrene is that we've doped it with a ferromagnetic nanoparticle so that we can attract it with a magnet. But that's not how we get these little discs out of the array. It's shown here on the inset on the right is one of these discs that have been partially removed. This microwell portion of the array is made out of this material called polydimethyl siloxane, or PDMS. It's like a soft plastic, and we take a needle and stick it through the base of the array and push out the Microraft, our cell carrier. The cell stays attached to it. We bring in a magnet, pick up that raft, and deposit it into a secondary container. So the technology has gone through a number of different form factors. We have a manual system. We have a couple of different automated systems. One is attached to a standard epifluorescence inverted microscope. The one I'm going to show you here is a wide field automated system so that we can image a large portion of the array over time. I'm going to start off. This is a animation. We see our array right there in the middle, a release device that has the needle, microscope objective above. Our magnetic wand is here that picks up the release raft, and we plate it into a 96 well plate. So if you look at this under a microscope, you see a target cell shown here in green, and then the little needle comes up, releases the raft, in comes our magnet, picks up that raft, and then takes it to another, the 96 well plate, and drops it into that well, and just does that iteratively. Point out that all of this is an automated system. The image analysis is automated. The technology to move these into the next well is automated. So it makes it a simple system to use in reality. Now, you can imagine you'd use selection criteria or some fluorescent signature of the cell to identify which ones are your target cell. Here we've wanted to identify cells that were expressing GFP histone so that they would have fluorescent nuclei. Cells are plated down at time 0, allowed to grow into a small colony in this case. And if you look in the upper panel A, the bright field shows 9 of the 45,000 sites. Most of the cells most of the rafts have cells in them, but only this one in the center has 3 cells, which are each expressing the histone GFP. We release that, collect it. You can see they still cells are still present, and then we can expand those into a clonal colony. But you could do that with FACS. You could do that with limiting dilution. What kind of makes this technology powerful and unique is the ability to image those cells over time and employ temporal characteristics as your selection criteria. So what I'm going to show you now is the ability to sort out efficient killing cytotoxic T lymphocytes. Our collaborator is interested in understanding better what makes a good CTL for immunotherapy. It's CAR T therapy and others. So we're interested in the target cell being tumor cells. We're working on this at the present time. The data I'm going to show you is a developmental experiment to develop a cytotoxic assay for our system. The target cells in this case are dendritic cells that have been primed with a flu peptide, and then the CTLs have been raised against that flu peptide so that they would be specific for the target cells. The approach we take is to take our dendritic cells, which are stained in blue, and I'll show you a movie of this in just a minute, and plate many target cells per well on the array. Then we plate one CTL in each well. Again, we use Poisson distributions to place the cells, so many of the wells will be empty. Those that have a cell, a CTL, will just have one. So that CTL will be red. And then in the media surrounding the cell is a live dead indicator called cytox green. So when the cell dies, the target cell dies, it will turn green. So then the idea is to measure cytotoxicity over time and then sort out the efficient killers and understand better what their biology is. So in the schematic on the left, it just shows kind of a cross-section view of a couple of different wells that show wells that have target cells, other wells that have target cells in one CTL, and then in the next panel below it, you can see that in one case, all the target cells have been killed, very efficient CTL, whereas in other panels, the CTL has been less efficient. And so now in the upper right section here, can't get my, there it goes. We'll have a time resolved a movie over a 6-hour period of a CTL assay. And we're just looking at one well of the many thousands on the array. In this, oh, yeah. There we go. So as time goes on, you'll see our little red CTL interacting with its target cells and the target cells turning green. Now it's replaying. So let me move on to the next panel. So what we've done is gone through and imaged that array over a 6-hour period, analyzed the data, found array wells with multiple dead target cells, and collected those wells, giving us our CTL. The CTL is then expanded in culture so that we can verify that it is specific killing for the flu peptide that has been loaded into the dendritic cell. We used tetramer-based cytometry, and indeed showed that in the case of the CTLs that were the most efficient killers, they were specific for the proper flu peptide, not the control peptide. So a lot of work has been done with this. I won't go into all of it. Just point out some of the capabilities of cell sorting with this technology. One is the high viability that I mentioned before. Makes it very appropriate for using in primary cells. Small sample sizes don't upset us. You can take fine needle aspirate with fewer than 1,000 cells and do a sort on that. That's something that you simply can't do with FACS. You can look at the morphology of the cell, particularly the adherent cells that attach to our little rafts, and you can pick out, we've had collaborators who want to pick out cells based solely on their morphology with that fluorescence. But I think more importantly is the power of being able to add in temporal selection criteria. I showed you the cytotoxicity assay as one example. Another example is collaborators who wanted to look at cells based on their doubling time. And there are other options along those lines as well. So we're seeing people use this for cloning, particularly genetically engineered cells. We have seen a lot of people interested in using this as a workflow to get their single cells for single cell RNA sequencing. We even have one collaborator who's interested in using this for intestinal organoids, shown in that panel on the far right. And that is a segue into my next topic, which is our work on organ on a chip. As I mentioned earlier, this is a growing area where people are making gut on a chip, heart on a chip, lung on a chip, just about every organ you can think of, either using cells from the target organ or using stem cell technology to create those organs on a microfabricated device. That's the chip part. Our work is primarily or solely on the intestine on a chip. I like to call it gut on a chip because it just has a better ring to it. And there's a lot of potential applications for this technology. You know, the intestinal epithelium, which is the portion of the intestine that we work with, is a highly stem cell driven tissue. The shown here in the upper left panel is immunohistochemical staining of a piece of intestinal tissue showing the intestinal crypt. At the base of the crypt are stem cells, which are proliferative and self-renew, but then there's daughter cells go on to differentiate, and those differentiated cells undergo apoptosis in 5 to 7 days. And so basically the entire lining of your intestine turns over every week. So the stem cells are very highly proliferative. There's a lot of interest in understanding the stem cell behavior for regeneration and other properties. But there's also a very strong interest in the pharmaceutical industry for studying transport and metabolism of drugs across the intestinal epithelium. The gut, the intestine, is one of the richest sources of hormones in our body. Recently, microbiome, the gut of the gut, has been a very hot topic. So microbiome studies are important in understanding how microbiome interacts with our body. And of course, disease models are of interest, such as inflammatory bowel disease. And all of these areas are areas that we are working towards. So we started this shortly after a group out of the Netherlands, Hans Clevers, developed an approach for keeping the intestinal stem cells in prolonged culture. They figured out how to get the stem cells to self-renew as well as differentiate into all of the lineages found in the intestinal epithelium. The problem was that the approach was to bury the stem cells in a patty of matrigel and then adding growth factors and other factors to the media overlying the matrigel. And what you ended up with is this complex three-dimensional spheroidal structure you see in this color photograph. And the shortcoming with this is that the lumen is inside that structure, so it's very difficult to access. Because of the complexity, it's difficult to image and quantify what's going on with the stem cell renewal and differentiation. You can't measure transport in a structure like this. And it's not a high throughput for screening, which is something that the pharmaceutical industry would like to have. Also, the cells are just randomly distributed, so there's no stem cell niche like you have in the crypt, and differentiated cells against the lumen. So we worked for a couple of years trying to develop a two-dimensional monolayer system so that we could basically recapitulate the organoid in this two-layer format, because that would resolve many of the issues with the organoid system. So what we did after much work is shown here in schematic form. This is a single well of a multi-well plate out here, and there is a transwell insert in this well. We have plated a hydrogel scaffold, we call it a biomimetic scaffold or synthetic lamina propria, on the porous membrane of the transwell plate. And then we plate our stem cells on top of that scaffold. We add in the media above and below the factors that will help support stem cell self-renewal and differentiation. The stem cells will spontaneously differentiate into all the lineages of the intestinal epithelium. And then that provides us with a two-dimensional monolayer system. And I will show you some data from that in just a moment. One of the questions I often get is, where do you get the stem cells? I should tell you that these are not true stem cells in the crypt. they've already undergone some epigenetic modifications that they will only differentiate into the lineages of the epithelium. They won't differentiate into muscle, nerve, so they're not pluripotential, but that actually makes our job a lot easier. So we get these stem cells from either transplant grade organs or surgical specimens. Shown here in the 15 ml tube are 2 pinch biopsies. And it's surprisingly easy to get isolated crypts in pretty pure form out of that tissue. Shown here are several crypts that are in suspension. Get my pointer. The stem cells are located right here in the base of these crypts. They're the only proliferative cells in this form. And so we plate them on our biomimetic scaffold and expand the stem cells, and then we can maintain them as cell lines. They can be frozen down, biobanked, and then taken out in future times at your convenience. Generally, we found that with human stem cells, we can go for at least 15 to 20 passages without seeing any karyotypic abnormalities. There's some data out that says that there might be some more subtle mutations that occur out at 15 to 20 passages. So we generally limit our passage numbers to 10 to 12 for our experiments. So our 2D monolayer has the same and appropriate cell types as you see in the organoid system. In these images here, we see these are patches of cells, not individual cells, patches of cells, and they've been stained in this case with a proliferative cell marker called EDU. You'll see that through in further slides. And honed down on one of these patches, you can see that there are proliferative cells and non-proliferative cells, cells that have undergone spontaneous differentiation into various cell types. They also, our stem cells also express SOX9, which is a marker for stem and progenitor cells, the proliferative cells in the crypt. And we have goblet cells shown here with staining for mucin or MUC2, and we have enteroendocrine cells. These are the hormone-producing cells of the gut. If it's small intestine, we have Paneth cells, and if it's I don't show that here. But if we look at our monolayer and side view, we can see that cells are columnar, and they are, or I should say cuboidal would be a more appropriate description here. The cells nuclei are stained blue in these panels. In the first panel, villi is a marker for the brush border microvilli. Second panel shows another marker for the brush border, actin, with integrin beta 4, which is expressed along the basal aspect, and that's against our biomimetic scaffold as is appropriate. And then on the far right side, the third panel just shows a cytoplasmic marker, sodium potassium ATPase, which is present in the cytoplasm of the cells. And if you let these patches grow, they'll grow into a confluent monolayer. They'll develop tight junctions as they differentiate. And shown here are some tight junction proteins, ZO1 and occludin. Won't get too much into it here, but developing an impermeable monolayer is important for studying transport in these two-dimensional monolayers. Speaking of transport, we do have transport proteins present at both the mRNA and protein level, and their expression levels are very consistent with what you see in vivo tissue. We also have metabolic enzymes present. Won't get into too much of our work on these two areas. This is mostly of interest to pharmaceutical companies that are looking at using this technology for drug screening. We'll talk a little bit about our work with inflammatory cytokines. The gut epithelium makes IL-8 particularly, and our monolayers do as well. Interestingly, if we activate the cells at baseline with no activation, we see a significant amount of IL-8 production, but it's all towards the luminal side of the monolayer, very little in the basal side. We do ELISA measurements to measure the concentration of IL-8 in the luminal and basal media. However, if you stimulate these cells with TNF, you see an interesting directional phenomena, and that is if you add the TNF to the lumen, you see maybe a little bump in IL-8 production, but very little production into or excretion into the basal fluid. However, if you add the TNF basally, you get a significant amount of IL-8 production into the what would be the bloodstream, so to speak. So without going into a lot of hand waving about what this means, it is thought to be possibly related to sentinel effect, where you get IL-8 production if you have some damage to the epithelium, and bringing in inflammatory cells and fibroblasts to help repair and protect you from any bacteria that might encroach. Moving on to a set of experiments that we're doing now. We're developing a variety of screening assays for looking at the effects of dietary compounds on hormone production by the gut. You know, the intestine is probably the largest or one of the largest producers of hormones. Of particular interest because of its role in the gut-brain axis, and satiety is serotonin, or 5-hydroxytryptamine. And shown on the left upper panel is serotonin-containing enteroendocrine cells. I see our time is starting to get close, so let me just kind of fly through these next few slides. We screened 13 dietary factors and saw 2 big hits. The tall histogram here is cinnamonaldehyde from cinnamon, and the next big hit was wasabi, allyl isothiocyanate. And that is of interest to people in the nutrition industry looking for various compounds and how they may interact with the hormone production by the gut. You know, this has all been done with our 2D monolayer systems, what I've talked about so far, but wouldn't it be great if we could make a 3D model? Really quickly, you know, the crypt is a 3D polarized structure with differentiated cells at the luminal aspect and our stem cell niche at the base. And the polarization is thought due to chemical gradients of growth factors. Wnt, Notch, arsphonin are growth factors that promote stem cell self-renewal, proliferation, and morphogenic factors like BMP, bone morphogenic protein, promote differentiation. But if you look closely at these structures, they're really like a folded monolayer. There's a monolayer of cells that line the crypt, and the crypt interior is open, filled with mucus generally. So we thought that perhaps we could take our synthetic lamina propria, that biomimetic scaffold, and make a 3D architecture that mimicked the crypt, then plate our stem cells and create a continuous monolayer that was folded into the invaginations. Then we would apply a chemical gradient of growth factors, and maybe we could get a proliferative compartment at the base of the crypt and differentiated cells up at the lumen. And make a long story short, it worked. This image is some of our in vitro crypts that have been removed from the scaffold and stained with EDU, which again stains the proliferative cells that you can see those at the base, whereas a differentiated cell marker, alkaline phosphatase, that marks enterocytes, is at the lumen. The crypts are appropriately polarized based on EDU staining, And if you look closely at one of these, they're filled with mucus. So we make, arrays. That's what we kind of are good at. So we made, in this case, an array of 110 of these colonic crypt-like structures. Looking at them from above, it's a little hard to make heads or tails of this, but basically you see that the EDU stained proliferative cells are all located within the crypt, and the differentiated marker alkaline phosphatase is present over the lumen. If you take one of these luminal sections and look at it in cross section, similar to what I showed you earlier, you can see in the bottom, Esrin is another stain for microvilli, and the cells are cuboidal with appropriate polarization. Now we're talking about polarization of the single cell layer, not of the crypt itself. Using a little more sophisticated staining method, this is one of our crypt arrays. Crypts have been taken out of the array and stained with a differentiated marker KRT20, as well as stem cell marker OLFM4, showing how the crypt is polarized. And if you put that against the immunohistochemistry example that I showed at the very beginning of this portion of the talk, you can see there's a great deal of similarity. Given the time, I won't go into a lot of detail, but we use this to screen the impact of short-chain fatty acids on our crypts. Short-chain fatty acids are the metabolic byproduct of fiber fermentation by microbiota in your gut. Acetate, propionate, and butyrate are the three we studied. Butyrate is felt to be a cancer preventive, and what we showed in this experiment is that of the three, butyrate had the most potent impact on reducing proliferation and enhancing differentiation in this system. I want to really quickly touch on our co-culture systems for microbiota. I think that's going to be a really big topic in the coming years. We started off with just taking our monolayer system and adding probiotics, in this case Lactobacilli rhamnosus, and then realized when we're looking at this, these are SEM images, there's something wrong here, and that is that the bacteria are right up against the cell. In the gut, particularly in the colon, there's a thick layer of mucus that separates the bacterial content of the lumen from the cells. And we went back to the drawing board, developed techniques to generate a thick layer of mucus, shown here in this panel, with our cells along the bottom, the mucus 300 microns thick, separating EGFP expressing E. coli. So we think that's going to be important as we try to do these co-culture experiments. But there's another aspect that we have to deal with, and that is the fact that particularly in the colon, you have an anaerobic, oxygen-free environment. And many of the microbiota that are present in the microbiome of the colon are obligate anaerobes. Even short-term exposure to room air, oxygen will cause them to die. So we had to go back and redesign our system in order to allow us to generate a reservoir in the lumen that's anaerobic while maintaining oxygen exposure to our human cells, which need oxygen to survive. And so up in the upper left-hand corner here is a computer model of the technique, the design that we chose. In the blue is low oxygen, 0 oxygen. The red is high oxygen. Our cells sit right here. This is the border between the luminal region and the basal reservoir. We expand that to look at here. We see that we can generate by computer an oxygen gradient that will provide oxygen to the stem cells and differentiate cells of the crypts and maintain a anaerobic reservoir. In the right panel, upper panel, we have designed a little cassette for this. long story short, we can use the cells themselves, not the microbiota, the human stem cells, cells, to deoxygenate the luminal compartment and then add our microbiota into that after the deoxygenation step without, it happens spontaneously. So it makes life pretty easy. Right now, we've shown that we can grow obligate anaerobes in this system with our cells. Our goal is to get up to a 7-day co-culture system, and that's where we are with that right now. So I think that ends my talk. A lot of great collaborators at UNC. Hopefully, as I said, we'll develop a network similar to that here. And of course, we have been fortunate to be supported by the National Institutes of Health over the years. So with that, I will escape and take any questions. Let me just, I don't know why it's saving. Okay, Q&A. No open questions.
Yeah, so I don't know, Laura, whether you want us to send the questions on the Q&A or by microphone, but anyway, let me start while people are thinking. So, have you, Chris, have you looked at the immune cell interactions? I think you have a publication with IVD and looking at immune cell interactions with the gut on a chip.
The answer to that is no. We have co-cultured peripheral blood monocytes with the IL-8 work, but that's just some preliminary data. We're currently working with a group in, or starting to work with a group here in GI to do exactly that, basically design a system where we have Peyer's patches, M cells, and start looking at inflammatory cells co-cultured with our system.
Okay.
We also have plans for being able to do culture of neurons, neuronal cells in the system as well, because that's also the gut-brain axis is a big area. Let's see.
At the moment, nothing for synovial or fibroblasts. You haven't started any work on that so far.
No. As I said, we kind of that ended when all my collaborators took off. So it'd be interesting to do it to do that project again. So I get one question here from Erica. One of the challenges of moving fibroblasts from in vivo to in vitro is that the hardness of the plastic activates fibroblasts. Are you able to modify the microrafts to reduce the hardness of the matrix to better mimic in vivo stiffness? We haven't done that, but the answer is yes, we can. Basically, that was the secret to our stem cell work, was to make that biomimetic scaffold the appropriate stiffness to support the stem cells and their response to the hardness or softness of the substrate. So what you would think about doing is coating the microrafts with a hydrogel of the appropriate stiffness to do what you want to do. We have actually done some invasion assays using a gel to watch cells as they invade into the hydrogel and through matrix metalloproteinase expression. Another question, do you have any experience working with neutrophils using your single cell sorting platform? No, I can't think that we certainly have not. I'm trying to think if some of our, if some others have used it. I haven't seen any papers published on that. You know, the technology is now commercialized by a company and a lot of people are using this outside of my realm, so I'm not sure. But I would expect that the cells, one of the points that you make is that the cells are very fragile and would it maintain good viability for this particular cell type? The answer I would suspect would be yes, because we've had stem cells, which are very, and other cell types, which are very fragile, and they survive the sort quite well. Any other questions? I will thank Tomas for his shout out that it was a good talk. Appreciate that, Tomas. Okay, well, I think that must be it. I don't see any other questions coming up. Oh, sorry. Tomas asked another question. How do you get fluorescent peptides into cells? Microinjection, penocytosis, cell-penetrating peptides. Some of the peptides are self-loading. Some of the lipids are self-loading. For instance, Sphingosine, we just incubate it with the cells.
Okay, well, if that's it, I want to thank you very much, Chris, for the terrific talk. I'm sure a lot of people will be interested in talking to you some more. So I think some of your information and contact information is on our website. Anyone wanting to contact Chris, just check out the information on the website. So thanks again.
Very good, Keith. Thank you.
Okay, bye-bye. We have some other, we follow on research, by the way, Chris, if you want to stay on, and we have 2 postdoctoral fellows who will be reporting on their research. So feel free to stay on if you would like.
Okay, I'll do that.
Good. Laura, you kind of take over? Laura? I guess maybe not. We need Laura to switch the controls.
Yeah, I just want to let Dr.